You'd think therapies that use light and electricity to heal our bodies would be the future of medicine. But what if we've been measuring them all wrong? A fascinating new paper suggests that to see if they really work, we shouldn't be watching the main event, we should be listening to the echo it leaves behind.
I mean, think about it. Using energy like light or electricity to heal sounds like something out of science fiction, and in some labs it actually works wonders. But there's this huge problem holding the whole field back, something scientists call a reproducibility crisis. So what does that really mean for the future of these treatments? Well, it means that a therapy showing incredible results in one lab might just completely fail in the next. This inconsistency has been a puzzle for researchers for years, and it's a major roadblock preventing these amazing ideas from becoming reliable treatments that you and I can actually use. For decades, the approach has been pretty straightforward. You apply a stimulus, like a pulse of light, and you watch for an immediate reaction, a simple cause and effect. But this new research argues that's like trying to understand an entire concert by only listening to the very first note.
And this is where the whole idea takes a radical turn. Instead of focusing on the moment of stimulation, what if the really important part, the real story, happens in the moments right after it ends? What if the true measure of success isn't the initial jolt, but the organized way the system recovers from it?
The best way to wrap your head around this is with a simple analogy. Think of a bell. Sure, you could study the physics of the hammer striking the metal, that's the stimulation. But the real character of the bell, what makes it special, is revealed in the sound it makes after being struck. Is it a clear, beautiful, long-lasting ring, or just a dull thud? That ring is what tells the real story. Okay, so how does this actually translate into a lab experiment? It's a simple three-step process you can see right here. First, scientists measure the system at rest. That's the baseline. Then, they apply the stimulation. And finally, and this is the most important part, they stop the stimulus and just listen. This washout phase is where they're trying to hear that ring. So that leads to the big question, right? How do you actually measure a biological ring? You can't just put a microphone next to a cell culture. You have to find a way to quantify that echo within the data itself. How do you tell the difference between a system that's just randomly returning to normal and one that's ringing with some kind of retained order? Well, the answer lies in a concept that the paper calls coherence. Now, in this context, coherence isn't some fuzzy, vague idea. It's defined by very specific, measurable qualities. How stable is the system's rhythm? How organized is its signal? And most importantly, how predictable is its behavior over time? A highly coherent system is, quite simply, an orderly one. And when we look at the data, we find something fascinating, a real paradox. Take a look at chart A on the left. The systems that got the active stimulation actually take longer to recover. But then, look at chart B on the right. That slower recovery contains much more structured, organized information. It isn't just fading away, it's relaxing in a very predictable, orderly fashion. This graph makes that point even clearer. It shows how accurately you can predict the system's next move. And the orange line, the stimulated system, has a much lower prediction error. That means its recovery is far more predictable and way less random. It's following a structured path back to normal. It is truly ringing. This measurable, structured ring is what the paper defines as the core finding, a specific signal they call the persistence signature. So, to find this signature, you first need to know what a boring recovery looks like. This is what's called the null relaxation model. It's the expected, simple decay curve you'd see if the stimulus had no lasting, organizing effect. Think of it as the dull thud of a cracked bell, just noise fading away into nothing. And here it is, the key finding, all visualized. That dotted orange line represents our boring thud, the expected ordinary decay. But the solid blue line is what the researchers actually observed. That shaded area between them, that is the persistence signature. It's the quantifiable proof of that structured ring, the echo of organization left behind by the stimulus. This quote from the paper really just sums up the entire shift in thinking. The question is no longer just, did the system react? The real question now is whether the recovery itself is special. Does it contain information? Is it more than just noise? Ultimately, this is more than just a new trick for data analysis. It really represents a fundamentally new way of understanding how these therapies interact with our biology. It's a whole new window. This final graph really pulls the whole story together. Just follow the orange line. You can see coherence is low at the baseline, then it shoots up during stimulation, just like you'd expect. But look what happens after the stimulation stops. It doesn't just crash back down. That coherence, that organization, persists deep into the recovery phase. That's the echo, right there in the data.
So why does all this matter? Well, it could finally solve that reproducibility problem by giving scientists a clear signature to look for, a new gold standard to measure success. It shifts the focus from a simple question of if a therapy works to the much richer question of how it helps the body recover and reorganize itself.
You know, this framework opens up a whole new way of seeing things. It moves us beyond just watching for a simple reaction and encourages us to listen for the complex, structured response that follows. And it leaves us with a pretty fascinating question. What other subtle echoes are happening inside our own bodies, just waiting for us to finally learn how to listen?